We are on our way out west of the city, leaving the urban and suburban area, and we're headed for those mountains that we see in front of us. That's White Tank Mountains Regional Park, which is home to one of our long-term established DesFert sites, as well as a number of other studies that happen out there. We're going to go through the booth, and now we're in the park. So as you can see, we're surrounded by Sonoran Desert. This is one of our rural sites, though you can see the city out there ahead of us over the horizon. And we'll just go around this curve here, and here we are at our research plot. So here we are at the White Tanks DesFert site, where things are a little brown because we've had such a dry monsoon season. The Desert Fertilization Experiment, aka DesFert, is one of our long-term studies where at sites both inside the city as well as outside where we are now, we experimentally add nitrogen or phosphorus or nitrogen and phosphorus together, and in comparison to control plots, we understand the fate and consequences of that fertilization for soil and plant nutrient content, plant growth and biomass, and community composition.
So this site receives CRS probes, stem elongation measurements, as well as foliar sampling, biomass and community composition for annuals, and also receives fertilization.
We also measure atmospheric deposition at this site using passive resin collectors, both under creosote and interplant spaces.
Today, I am going to demonstrate how we switch out our depth collectors. This is an interplant depth collector. I'm going to take 500 milliliters of water and pour it through the funnel, making sure to rinse the funnel, the screen, and the bird spikes. Once the water has entered the column and emptied through the column, I will switch it out with a replacement funnel. Funnel and empty through the column. I'm going to remove the collector. And replace it with a new one. Making sure to record the numbers that are going out and the numbers that are coming into the lab.
We do the same thing with the collectors that are placed under the canopy of creosote bushes. Together, they comprise the longest spatially and temporally explicit data set of nitrogen deposition in drylands, with data starting back in 1999 at some sites.
Stem elongation. Identify old tape. North. East. South. And west. Make measurement from node, inner node to inner node. Record. And done. Annuals biomass harvest. So you write site, date, subplot description, quad description, and initials. Take scissors, cut, bag. Roll up. Done. On to next.
The infrastructure of the Desperate sites provides the foundation for a lot of individual student projects.
Hi, my name is Anna Khan, and I just graduated with my Bachelor's of Science in Environmental Science at Arizona State University. And now I'm in a master's program at the Arizona State University Polytechnic campus. It's the ERM program, which stands for Environmental and Resource Management.
Students like Anna have been able to do independent research as undergraduate students as well as graduate students to understand the impacts of urbanization and fertilization on things like litter decomposition, photodegradation, and soil and vertebrate communities. As of March 2019, the white tanks are also part of Drought Net with the rainout shelters you see here. These structures passively intercept some but not all precipitation in the clear troughs, while still allowing for other natural inputs into the soil. The long-term data being generated allow us to understand the impacts of drought on vegetation not only within CAP but across the larger network of sites. To increase the U.S. Southwest's representation in Drought Net, we are doing this through a collaboration with colleagues at CAP LTER, including PhD student Tim Ohlert. And now we'll leave the white tanks and head to another moderately urbanized area within the CAP study area where we collect some of our long-term wildlife data.
Hi, my name is Jesse Lewis, and I'm an assistant professor working with the Adapting to City Life IRT and Parks and River IRT, where I'm a co-lead with a CAP LTER. And in collaboration with undergraduate and graduate students in my lab, as well as colleagues with CAP, we evaluate the effects of urbanization on a diversity of wildlife species, including bats, scorpions, a suite of terrestrial mammals and birds. And in particular, we use remote wildlife cameras, such as this one featured here, placed across the urban gradient from downtown Phoenix to our remote open space parks, such as the White Tank Mountains Regional Park and along the Salt River, to evaluate the effects of urbanization on populations, species interactions, and community composition. And these projects are ongoing, but preliminary results demonstrate that some species are urban avoiders, only being found in undeveloped areas, whereas other species are urban exploiters and able to take advantage of resources in our most urban environments. So we look forward to following up again in the near future and sharing more results as these projects continue, which we're really excited about. So thank you very much.